Microbes in human welfare. Besides microscopic plants and animals, microbes are the major components of biological systems on this earth. You have studied about the diversity of living organisms in Besides macroscopic plants and animals, microbes are the major components of biological systems on this earth. You have studied about the diversity of living organisms in class 9. Do you remember which kingdom among the living organisms contain microorganisms? It is Monera, Protista, fungi, and plantae. Which are the ones that are only microscopic? These are Monera and Protista. Microbes are present everywhere in soil, water, air, inside our body, and that of the other animals and plants. They are present even at sites where no other life form could possibly exist. Sites such as deep inside the geyser, that is thermoacidophils, thermal vents, where the temperature may be as high as 100 degrees Celsius, deep in the soil, under the layers of snow several meters thick, and in highly acidic environments. Microbes are diverse protozoa, bacteria, fungi, and microscopic animals and plant viruses, viroids, and also prions that are proteinaceous infectious agents. Some of the microbes are shown in figure 8.1 and 8.2. Microbes are diverse protozoa, bacteria, fungi, microscopic animals, and plant. Microbes like bacteria and many fungi can be grown on nutritive media to form colonies that can be seen with naked eyes. Such cultures are useful in studies on microorganisms. Diagrams. First diagram is of rod shaped magnified bacteria, which is bacillus. Second is spherical shaped magnified bacteria, cocci shaped. And third one is a rod shaped bacillus bacteria with a flagellum, which is flagellated bacillus. Then we have the second 8.2 figure 8.2. First one is a bacteriophage. It's also in 11th standard NCERT. Just we have head, collar, tail, plate, pins, and prongs. Then we have adenovirus, which is a causative agent for viral pneumonia, which causes respiratory infections. And then we have a rod-shaped tobacco mosaic virus, TMV virus. It is a plant, which is plant virus, and it has ssRNA. Then we have colonies of bacteria in a petri dish, fungal colony growing in a petri dish. In chapter 7, you have read that microbes cause a large number of diseases in human beings. They also cause diseases in animals and plants. But this should not make you think that all microbes are harmful. Several microbes are useful to man in diverse ways. Some of the most important contributions of microbes to human welfare are discussed in this chapter. Microbes in household products. You would be surprised to know that we use microbes or products derived from them every day. A common example is production of curd from milk. So I need to write it down here. Write it down here. Calorie. Oxidation reduction. Okay. Mhm, mhm, mhm. Should be somewhere here. Got it. So first we have lab, which is called as lactic acid bacteria. कर्ड मेकिंग में यूज होता है और समथिंग सो फर्स्ट थिंग वी हैव इज lab, which is lactic acid bacteria. कर्ड बनाने में यूज होता है. विटामिन B12 एनरिच करता है and the bacteria specific for this purpose is responsible for this specific purpose is pseudomonas denitrificans. Pseudomonas denitrificans. तो ये क्या करता है? पार्शियली डाइजेस्ट एंड कोआगुलेट मिल्क प्रोटीन. Partially digest and coagulate milk protein. Then next we have Saccharomyces cerevisiae. It is called as brewer's yeast, used in the production of ethanol, also for bread making and as invertase enzyme. Yeast इसको बोलते हैं फंगा है ये एक. कौन सा वाला फंगा है? एस्कोमाइसिट्स. और ये क्या है? Saccharomyces cerevisiae. Saccharomyces cerevisiae. Then we have cheese of 3 types. Then toddy तो पता है, traditional drink used by South Indian by fermenting sap plants. Ferment fish, soybean, and bamboo. Cheese is one of the oldest items in which microbes are used. So 3 types of cheese: Swiss, Camembert, Roquefort. Swiss cheese, Propionibacterium shermani. बैक्टीरिया से होता है. Large holes. Then we have Camembert cheese, Penicillium camemberti. Penicillium camemberti. It is a fungi. And Roquefort cheeses, Penicillium roqueforti. Penicillium roqueforti. It is also a fungi. Swiss cheese is derived from a bacteria. Uh, then we have other acids like citric acid and gluconic acid derived from Aspergillus niger. It is a fungi used to prepare citric acid, gluconic acid. Citric acid is vitamin C. Then we have acetic acid. Acetic acid के लिए Acetobacter aceti. Acetic acid या vinegar we can say Acetobacter aceti. Bacteria तो ये bacteria है. And then we have another butyric acid, Clostridium butylicum. Butylicum. This is also a bacterium. Now we have streptomycin antibiotic. Streptomycin 3 टाइप्स के होते हैं एरिथ्रोस, ग्रेसियस और वेनेजुएली. एरिथ्रोमाइसिन, स्ट्रेप्टोमाइसिन एरिथ्रियस से जो मिलता है उसको एरिथ्रोमाइसिन, ग्रेसियस से मिलता है स्ट्रेप्टोमाइसिन और वेनेजुएला से मिलता है क्लोरोमाइसिन. देन फ्लेमिंग was first to मतलब उन्होंने इन्वेंट किया था एंटीबायोटिक्स पेनिसिलियम नोटाटम से. उन्होंने क्या देखा था कि सिफाइलोकोकाई बैक्टीरियम की जो कॉलोनीज है दैट डिड नॉट ग्रो अराउंड पेनिसिलियम नोटाटम which led to the discovery of antibiotics. Further, Florey and Chain क्या बोलते हैं उसको gave out like discovered the true potential, full potential and effective antibiotic was established by Ernst Chain and Howard Florey in 1945 World War II के टाइम. World War II in 1945. उसके बाद we have other things like lipase, protease, clotbuster, streptokinase, and cyclosporin, immunosuppressor cyclosporin is tannins, that is blood cholesterol lowering agents. 5 things we have: lipase, protease, streptokinase, immunosuppressor, and blood cholesterol lowering agents. Lipase are stain removal detergent. Rhizopus spp से मिलता है ये बैक्टीरिया से. So ये बैक्टीरिया से मिलता है बैक्टीरिया. Pectinase and protease are used to clarify juices and juice clarifiers and obtained from fungi. Obtained from fungi. Then we have streptokinase, clot buster, streptococcus bacteria. Immunosuppressive agent cyclosporin A, and it is a cyclosporin A, Trichoderma polysporum, और ये fungi से आता है. Cyclosporin A Trichoderma polysporum, fungi. Clotbuster, streptokinase, streptococcus, bacteria से आता है. Blood cholesterol lowering agent stannin, and it is obtained from Monascus purpurus, which is again a fungi. Which is a fungi. तो फंगाई 3 फंगाई कौन सा? एक पेक्टाइनेज एंड प्रोटीज वाला, एक इम्यूनोसप्रेसिव एजेंट साइक्लोस्पोरिन ए, एंड एक है ब्लड कोलेस्ट्रॉल लोअरिंग एजेंट स्टैनिन्स. All these three are fungi. Then we come to biogas. Anaerobic digestion, solubilization और hydrolysis first step होता है, जिसमें insoluble polymer in the presence of bacterial hydrolytic enzyme is converted into simple soluble monomer. Now actinogenesis या acidogenesis होता है. Acetogenesis या acidogenesis होता है, in which monomer is converted into as organic acid, to be specific acetic acid, in the presence of acidogenic bacteria. Now, then we have the last step, which is the most important step, the useful step we should say, जिसमें organic acid है, जो कि मतलब हमारा acetic acid, it is converted into methane plus carbon dioxide plus other gases in the presence of methanogenic bacteria such as methanococcus and methanobacillus. So first step is anaerobic digestion. Anaerobic digestion, उसके बाद एसिडोजेनेसिस, और फिर उसके बाद आता है मिथेनोजेनेसिस. एनारोबिक डाइजेशन में हम यूज करते हैं हाइड्रोलाइटिक एंजाइम्स. एसिडोजेनेसिस में हम एसिडोजेनिक बैक्टीरिया यूज करते हैं. मिथेनोजेनेसिस में मिथेनोजेनिक बैक्टीरिया. This is all for the chapter. Now we'll quickly read down the NCERT. Microbes in human welfare. In chapter 7, we have read that microbes cause a large number of diseases in human beings. They also cause diseases in animals and plants. But this should not make you think that all microbes are harmful. Several microbes are useful to man in diverse ways. Some of the most important contributions of microbes to human welfare are discussed in this chapter. Page number 150, there are some diagrams. First one is of Bacillus actinoscoccus. Third one is of flagellated Bacillus bacteria, a rod-shaped bacterium showing flagella. Then we have viruses. First one is the bacteriophage. Tobacco वाला था, नहीं bacteriophage नहीं है, जिसमें head, collar, tail, plate, wings और prongs. B वाला है adenovirus, which is just the hexagonal things like that, which causes respiratory infections. And third one is the rod-shaped tobacco virus, that is the DMD virus, which causes disease in tobacco plant, which has ssRNA. Then the other figure has colonies of bacteria growing in a petri dish, and on the colony growing in a petri dish. Microbes and household. You would be surprised to know that we use microbes for products derived from them every day. A common example is the production of curd from milk. Microorganisms such as lactobacillus and other formerly called as lactic acid bacteria lab grown in milk and convert it to curd. During growth, the lab produce acids that coagulate and partially digest the milk proteins. A small amount of curd added to fresh milk as inoculum or starter contain millions of lab, which at stable temperatures multiply, thus converting milk to curd, which also improves its nutritional quality by increasing vitamin B12 in our stomachs. The lab play a very beneficial role in checking disease-causing microbes. So lab has three works. First is it partially digests. We'll read it again. Convert it to curd during the growth of lab. It coagulates and partially digests the milk proteins. It partially digests the milk
proteins, small molecule starter, milk convert the hecker to milk to then it increases the B12 vitamin B12 nutritional quality, and in our stomach it checks disease causing microbes. The dough which is used for making foods such as dosa and idli is also fermented by bacteria. The puffed up appearance of dough is due to the formation of carbon dioxide gas. Can you tell which metabolic pathway is taking place resulting in the formation of carbon dioxide? Where do you think bacteria for these fermentation come from? It's fermentation. Okay? Similarly, the dough which is used for making bread is fermented using baker's yeast, that is Saccharomyces cerevisiae. A number of traditional drinks and foods are also made of fermentation by the microbes. Roti is a traditional drink of some parts of southern India. It's made by fermenting sap from palms. Microbes are also used to ferment fish, soybean, and bamboo shoots to make food. Cheese is one of the oldest food items in which microbes were used. Different varieties of cheese are known by their characteristic texture, flavor, and taste. The specificity coming from the microbes used. For example, the large holes in Swiss cheese are due to the production of large amounts of by bacterium named Propionibacterium shermani. The Roquefort cheese are ripened by growing of specific fungi on them, which gives them a particular flavor. Penicillium roqueforti. And then we have Davenport cheese, is Penicillium davenporti. It is a fungi again. Microbes in industrial products. Even in industries, microbes are used to synthesize a number of products valuable to human beings. Beverages and antibiotics are some examples. Production on an industrial scale requires growing microbes in a very large vessel called fermenters. Fermented beverages. Microbes, especially yeast, have been used from time immemorial.
So this is important. इसमें से questions आता है. Even in industries, microbes are used to synthesize a number of products valuable to human beings. Beverages and antibiotics are some example. Production on an industrial scale requires growing microbes in a very large vessel called fermenters. Microbes, especially yeast, have been used from time immemorial for the production of beverages like wine, beer, whiskey, rum, brandy. For this,
For the production of beverages like wine, beer, whiskey, brandy, and rum. For this purpose, the same yeast Saccharomyces cerevisiae used for making bread and commonly called reverse yeast is used for fermenting multi cereals and fruit juices to produce ethanol. We recollect the metabolic reactions which result in the production of ethanol by yeast. It is called fermentation. Depending on the type of the raw material used for fermentation and the type of processing, with or without distillation, different types of alcoholic drinks are obtained. Wine and beer are produced without distillation, whereas whiskey, brandy, and rum are produced by distillation of the fermented broth. The photograph of fermented wine is shown in the figure. Antibiotics. Antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of human society. Anti is a Greek word that means against, and bio means like. Together they mean against and context of disease-causing organisms. Whereas, with reference to human beings, it is a pro-life and not against. Antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease-causing microbes. You are familiar with the commonly used antibiotic penicillin. Do you know that penicillin was the first antibiotic to be discovered and it was an chance discovery? Alexander Fleming, while working on Cephalococcus bacteria, once observed a mold growing in. You are familiar with the commonly used antibiotic penicillin. Do you know that penicillin was first discovered antibiotic and it was a chance discovery? Alexander Fleming, while working on Cephalococcus bacteria, once observed a mold growing in one of his unwashed culture plates around which Cephalococcus could not grow. He found out that it was due to a chemical produced by the mold and he named it penicillin after the mold, Penicillium notatum. However, its full potential as an effective antibiotic was established much later by Ernst Chain and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain, and Florey were awarded the Nobel Prize in 1945 for this discovery. After penicillin, other antibiotics were also purified from other microbes. Can you name some of the antibiotics and find out their sources? Antibiotics have greatly improved our capacity to treat deadly diseases such as plague, whooping cough, diphtheria, and leprosy, which used to kill millions all over the globe. Today, we cannot imagine a world without antibiotics. Chemicals, enzymes, and other bioactive molecules. Microbes are also used for commercial and industrial production of certain chemicals like organic acids, alcohols, and enzymes. Example of acids produced are asparagine agar, a fungus of citric acid; Acidobacter acidi, a bacterium of acetic acid; Clostridium butyricum, a bacterium of butyric acid. Microbes are also used for commercial and industrial production of certain chemicals like organic acids, alcohols, and enzymes. Example of acetic acid. Examples of asparagine agar, a fungus of citric acid; Acidobacterium acidi, a bacterium of acetic acid; Clostridium butyricum, a bacterium of butyric acid; and Lactobacillus, a bacterium of lactic acid. Gluconic acid, also produced by asparagine agar, and fumaric acid is produced by Rhizopus canicus. Yeast. Saccharomyces cerevisiae used for commercial production of ethanol. Microbes are also used in production of enzymes. Lipases are used in detergent formulations and are helpful in removing oily stains from the naughty. You must have noticed that bottled fruit juice brought from the market are clearer as compared to that made at home. This is because the bottled juice are clarified by the use of peptidases and proteases. Streptokinase, produced from the bacterium Streptococcus and modified by genetic engineering, is used as a clot buster for removing clots from the blood vessels of patients who have undergone myocardial infarction leading to heart attack. Another bioactive molecule called cyclosporine A that is used as an immunosuppressive agent in organ transplant patients is produced by a fungus called Trichoderma polysporum. Statins produced by yeast, Monascus purpureus, have been commercialized as blood cholesterol lowering agents. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So, to sum it all, lactic acid bacteria, curd, B12, bacterium Pseudomonas nitrificans, Saccharomyces cerevisiae, yeast, reverse yeast, ethanol, bread making, invert AIDS enzyme, cheese, Roquefort cheese, Penicillium roqueforti, a fungi, Camembert cheese, Penicillium camemberti, a fungi, Swiss cheese, Propionibacterium shermani, a bacterium, citric acid, gluconic acid, aspergillus niger, fungi, vitamin C, acetic acid, Acidobacter acidi, bacterium, vinegar, acetic acid, Acetobacter acidi, butyric acid, Clostridium butyricum bacteria, Streptomycin bacteria, Fleming ने penicillin bacteria, Cephalococcus को grow नहीं करने देना. So it is made from Penicillium notatum and it does not allow the growth of Cephalococcus. Lipase, detergent Rhizopus spp, a bacteria, pectinase and protease, juice clarifiers, Streptococcus libertine, a fungi, Streptokinase, clot buster, Streptococcus bacteria, cyclosporine, suppressor, Trichoderma polysporum, fungi, statins, blood cholesterol lowering agent, Monascus purpureus, a fungi. It increases the manufacturing process of cholesterol in the body by competitive inhibition. Biogas production, three steps: anaerobic digestion, solubilization, or hydrolysis. Insoluble polymer. In presence of bacterial hydrolytic enzyme is broken into simple soluble monomers. Then acidogenesis is performed where the monomer is treated with acidogenic bacteria to produce an organic acid, preferably acetic acid. The organic acid thus formed, that is acetic acid, is treated with methanogenic bacteria to form biogas, which is methane carbon dioxide principal gases. Methanogenic bacteria, example Methanococcus and Methanobacillus. Microbes in sewage. You know that large quantities of wastewater are generated every day in cities and towns. A major component of this wastewater is human excreta. This municipal wastewater is also called sewage. It contains large amount of organic matter and microbes, many of which are pathogenic. Have you ever wondered where this huge quantity of sewage or urban wastewater is deposited or daily? This cannot be discarded into natural bodies, water bodies like liver and rivers and streams, and you can understand why. Before disposal, hence sewage is treated with sewage treatment. Sewage is treated in sewage treatment plants (STPs) to make it less polluting. Treatment of wastewater is done by heterotropic microbes naturally present in the sewage. This treatment is carried out in two stages. Treatment of wastewater is done by heterotropic microbes naturally present in the sewage. This treatment is carried out in two stages. Primary treatment. These treatment steps basically involve physical removal of particles, large and small, from the sewage through filtration and sedimentation. These are removed in stages. Initially, floating debris is removed by sequential filtration. Then the grit, soil, and small pebbles are removed by sedimentation. All solid that settles form the primary sludge, and the supernatant forms the effluent. The effluent from the primary settling tank is taken for secondary treatment. Secondary treatment or biological treatment. The primary effluent is passed into large aeration tanks where it is constantly agitated mechanically and air is pumped out into it. This allows vigorous growth of useful aerobic microbes into flocks. Masses of bacteria associated with fungal filaments to form mesh-like structure. While growing, these microbes consume the major part of organic matter in the effluent. This significantly reduces the BOD, which is the biochemical oxygen demand of the effluent. BOD, biochemical oxygen demand. It is biochemical oxygen demand. Refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. The sewage water is treated, the BOD is reduced. The BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus indicate indirectly BOD is a measure of the organic matter present in the water. The greater the BOD of the water, more is its polluting potential. Once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into a settling tank where the bacterial flocks are allowed to sediment. This sediment is called as the activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as the inoculum. The remaining major polluting potential. Once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into a settling tank where the bacterial flocks are allowed to sediment. This sediment is called as
While growing, these microbes consume the major part of organic matter in the effluent. This significantly reduces the BOD, that is the biochemical oxygen demand of the effluent. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. Kitna liter? 1 liter of water. BOD refers to the amount of oxygen that would be consumed in if all the organic matter were oxidized by bacteria. क्या करा था? Oxidized. The sewage water is treated till BOD is reduced. BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus indirectly BOD is the measure of organic matter present in the water. The greater the BOD of the wastewater, more it is polluting potential.
activated sludge. A small part of the activated sludge is pumped back into the aeration tank to
Sorry. Once the BOD of the sewage or wastewater is reduced significantly, the effluent is then passed into a second settling tank where the bacterial flocks are allowed to sediment. This sediment is called activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as the inoculum. The remaining major part of the sludge is pumped into large tanks called anaerobic sludge digester. Here, the other kind of bacteria grow anaerobically, digest the bacteria and the fungi in the sludge. सेकेंडरी ट्रीटमेंट में यूजफुल एरोबिक माइक्रोब्स और उसके बाद एक्टिवेटेड स्लज को हम anaerobic bacteria में डालते हैं anaerobic sludge digester. Here the kind of bacteria digest the bacteria and the fungi in the sludge. During digestion, bacteria produce a mixture of gases such as methane, हाइड्रोजन सल्फाइड एंड कार्बन डाइऑक्साइड. ये कहाँ पे होता है? एनाएरोबिक स्लज डाइजेशन में होता है. These gases form biogas and can be used as source of energy as it is inflammable.
as the inoculum. The remaining major part of the sludge is pumped into large tanks called anaerobic sludge digesters. Here, other kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. During this digestion, bacteria produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide. These gases form biogas and can be used as source of energy as it is inflammable. Secondary treatment, agitation, flux formation, and aerobic microbes settling and flux settling activated sludge. Anaerobic sludge digester anaerobic bacteria digest bacteria and fungi in sludge. The effluent from the secondary treatment plant is generally released into natural water bodies like rivers and streams, and aerial flow of such plant is shown. You can appreciate how microbes play a major role in treating millions of gallons of water, wastewater every day across the globe. This methodology has been practiced for more than a century now in almost all parts of the world. Till date, no man-made technology has been able to rival the microbial treatment of sewage. You're aware that due to the increasing urbanization, sewage will produce much larger quantities than ever before. However, the number of sewage treatment plants are not increased enough to treat such large quantities. So the untreated sewage is often discharged directly into the rivers, leading to the pollution and increase in waterborne diseases. The Ministry of Education, the Ministry of Environment and Forests have initiated Ganga Action Plan and Yamuna Action Plan. The Ministry of Environment and Forests has initiated Ganga Action Plan and Yamuna Action Plan to serve these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants so that only treated sewage may be discharged into the rivers. A visit to a senior microbes and production of biogas. Biogas is a mixture of gases containing predominantly methane produced by the microbial activity, which may be used as a fuel. You have learned that microbes produce different types of gases and products during growth and metabolism. The type of the gases produced depends upon the microbes and the organic substance they utilize. In the example seated in relation to formulation of dough, cheese, milk, and production of beverages, the main gas produced was carbon dioxide. However, certain bacteria grow anaerobically on cellulosic material to produce large amount of methane along with carbon dioxide and hydrogen. These bacteria are collectively called methanogens, and such common bacterium is methanobacterium. These bacteria are commonly found in anaerobic sludge during sewage treatment. These bacteria are also present in the rumen, a part of stomach of cattle. A lot of cellulosic material present in the food of cattle is also present in the rumen. In rumen, these bacteria help in the breakdown of cellulose and play an important role in the nutrition of the cattle. Though you think human beings celebrate digest cellulose present in our foods, no. Thus, the excreta dung of cattle, commonly called co-penetrating these bacteria during dung can be used for generation of biogas, commonly called co-gas. The biogas plant consists of concrete tank 10 to 15 feet deep, in which bio wastes are collected, and a slurry of plant is fed. A floating cover is placed over the slab, which keeps on rising as the gas is produced in the tank due to the microbial activity. The biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. The spent slurry is removed through another outlet and may be used as fertilizer. Cattle dung is available in large quantities in rural areas where cattle are produced for a variety of purposes, so biogas plants are more often present in rural areas. The biogas thus produced is used for cooking and lighting. The picture of a biogas plant is shown in figure 8.8. The technology of biogas production was developed in India mainly due to the efforts of Indian Agricultural Research Institute (IARI) and Khadi and Village Industries Commission (KVIC). Indian Agricultural Research Institute and Khadi and Village Industries Commission (KVIC). India's school is microbes as biocontrol agents. Microbes and biocontrol agents. As biocontrol refers to the use of biological methods for controlling plant diseases and pests. In modern society, these problems have been tackled increasingly by use of chemicals. By use of insecticides and pesticides, these chemicals are toxic and extremely harmful to human beings and animals alike, and have been polluting our environment, soil, ground, water, fruits, vegetables, and croplands. Our soil is also polluted through use of weedicides to remove weeds. Biological control of pests and diseases. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduction of chemicals. A key belief of the organic farm is that biodiversity furthers health. The more the variety of landscape has, the more sustainable it is. The organic farmer therefore works to create a system where the insect that are sometimes called pests are not eradicated but instead are kept at manageable levels by complex systems of checks and balances within a living thriving ecosystem. Contrary to the conventional farming practices which often use chemical methods to kill both useful and harmful life forms indiscriminately, this is a holistic approach that seeks to develop an understanding of the webs of interaction between the myriad of organisms that constitute the field fauna and flora. The organic farmers hold the view that the eradication of the creatures that are often described as pests is not only possible but also undesirable, for without them the beneficial predatory and parasitic insects which depend upon them as food or host would not be able to survive. Thus, the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as the pests, and along their life cycles, patterns of feeding and the habitat they differ. This will help develop appreciable or appropriate means of biocontrol. The very familiar beetles with red and black markings, the ladybird and dragonfly, are used to get rid of aphids and mosquitoes respectively. An example of microbial biocontrol agent that can be introduced in order to control butterfly caterpillars is the bacteria Bacillus thuringiensis, often written as BT. These are available in sachets and dried spores, which are mixed with water and sprayed onto vulnerable plants such as brassicas and fruit trees, where these are eaten by the insect larvae in the gut of the larva. The toxin is released and the larva get killed. Bacterial diseases will kill the caterpillars but leave other insects unharmed. Because of the development of methods of genetic engineering in the last decade or so, the scientists have introduced B-glucogenesis toxin genes into plants. Such plants are resistant to attack by insect pests. BT cotton is one such example which is being cultivated in some states of our country. You will learn more about this in chapter 10. A biological control being developed for use in treatment of plant diseases is the fungus Trichoderma. Trichoderma species are free-living fungi that are very common in the root ecosystem. They are effective biocontrol agents for several pathogens, plant pathogens. Biocontrol developed for use in the treatment of plant disease is the fungus Trichoderma called this mycorrhizal. Baculovirus. Baculovirus are pathogens that attack insects and other arthropods. The majority of baculoviruses used as biocontrol agents are in the genus Nucleopolyhedrovirus (NPV). These viruses are excellent candidates for species-specific narrow spectrum insecticidal applications. They have been shown to have no negative impacts on plants, mammals, birds, fishes, or even on non-target insects. This is especially desirable when beneficial insects are being conserved to aid in an overall integrated pest management program or when an ecologically sensitive area is being treated. Microbes as biofertilizers, when our present-day lifestyles, with our present-day lifestyles, environmental pollution is a major cause of concern. The use of the chemical fertilizers to meet the ever-increasing demand of agricultural produce has contributed significantly to this pollution. Of course, we have now realized that there are problems associated with the overuse of chemical fertilizers, and there is a large pressure to switch to organic farming. The use of biofertilizers. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main sources of biofertilizers are bacteria, fungi, and cyanobacteria. You have studied about the nodules on the roots of leguminous plants formed by the symbiotic association of rhizobium, which is the bacteria. These bacteria fix atmospheric nitrogen into organic forms, which is used by the plants as nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil. Example, Azospirillum and Azotobacter, thus enriching the nitrogen content of the soil. Fungi are also known to form symbiotic associations with plants. Mycorrhiza, many members of the genus Glomus form mycorrhiza. The fungal symbiont in these associations absorb phosphorus from the soil and passes it to the plant. Plants having such associations show other benefits also, such as resistance to group 1 pathogens, tolerance to salinity and drought, and overall increase in the plant growth and development. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environment, many of which can fix atmospheric nitrogen, example Anabena, nostoc, ocellatorium, etc. In paddy fields, that is rice fields, cyanobacteria serve as an important biofertilizer. Blue-green alga also add organic matter to the soil and increase its fertility. Currently, in our country, a number of biofertilizers are available in commercial market and used to regulate soil nutrients and produce chemical fertilizers. Rhizobium symbiotic association, free living, Azospirillum and Azotobacter, bacteria fix nitrogen.